Assalamualaikum and hi, I am Nadiah together with my group mate Tuk Jahui would like to present about chromatography for pharmaceutical analysis. So these are our table of contents which consists of research topic, method and instrument being used, sample, instrument setting, result and also references. This is our first content, which is research topic, and the topic is the new TLC method for separation and determination of multi-component mixtures of plant extracts. Okay, next, on the right box, there are stated instruments that being used. The first one is 10 cm x 20 cm glass HPTLC plates coated with silica gel. Next is HPLC syringe, Teflon, Bunsen burner, Desaga, CD60 densitometer, essential oils, toluene, sulfuric acid, ethyl acetate and also vanillin ethanol solution. For the method on the left box, the first method is chromatography was performed on 10 cm x 20 cm glass HPTLC plates coated with silica gel. Then essential oils from juniper and thyme were obtained from Profam and dissolved in toluene to furnish 0.2% solutions. Next, the organic solvents such as toluene and sulfuric acid were purchased from Aventa Performance Materials, while the vanillin was obtained from MERCK, whereas anise aldehyde was received from Sigma Ulrich. The first step is 15 microliter of the toluene solutions of essential oils was supplied as 1 cm zone on the 10 x 20 cm chromatographic plates by means of the Hamilton HPLC syringe. Next, chromatographic plates after spot applications were developed in horizontal Teflon, DSL or DS111020 chamber in the first direction in traditional way with mixture of toluene and ethyl acetate as the solvent. The development distance were varied from 6 cm to 10 cm. Then the mobile phase has been evaporated. Then subsequently the plates was turned by 90 degrees Celsius and then separated zones were preconcentrated by development with a strong volatile solvent on the distance from 0.8 to 2 cm from the starting point. The chemical properties of investigated solutes influence the type of eluent applied. Then, the monoterpenes were developed with toluene or ethyl acetate, whereas more polar compounds require stronger eluent that is methanol or mixture water and methanol. The pre-concentration procedure was repeated until the zones were narrow and compact. After each repetition, the plate was dried. Then the plate was developed in full distance in the perpendicular direction, and all experiments were conducted at room temperature. The spots of separated compounds were detected with anise aldehyde in sulfuric acid, and then the plate was heated to 100 degrees Celsius until the color spots become visible. The spots were detected with the Desaga CD60 densitometer. These are the samples that are being used in this chromatography experiment. The first one is the 2 plants extract, which are from the plant of Juniperus and also Thymus. And then the next sample is silica gel.
The instrument setting for this new TLC method in separating and determining of multi-component mixture of plant extracts. There is a big difference for this method compared to the normal 1 TLC method. First is the preparations. In preparation, 10 cm x 20 cm of glass HPTLC plates coated with silica gel, 50 microliter of toluene solution of essential oils, which is the junipereolium and thymeolium, applied as a 1 cm zone on the chromatographic plates by using HPLC string. Secondly is the chromatographic development. In chromatographic development, the chromatographic plates after spot application were developed in horizontal with mixture of toluene and ethyl acetate as the eluent. The distance varied from 6 to 10 cm. Then let the mobile phase evaporate. After that, The plate was turned 90 degrees and the separated zones were pre-concentrated by development with a stronger volatile solvent on a distance of 0.8 to 2 cm from the starting point. The pre-concentration were repeated until the zone narrow and compact. After each repetition, the blade was dry. All of the experiments above were conducted at room temperature. Thirdly is the detection of compound. The spots of separated compounds were detected with anise aldehyde in sulfuric acid or with vanillin ethanol solution. The blade was heated to 100 degrees Celsius until the color spots became visible. Lastly, the spots were detected with the Saga CD60 densitometer. The next slide is the results and discussion. The first picture is the result separation of juniperoleum compounds, while the second picture the result of the separation of thymic-oleum. The A is by using the anise aldehyde sulfuric 4 acid reagent, and the B is by using the vanillin in ethanol reagent. This result shows that the new TRC method for separation and determination of multi-component mixture of plant extracts success. Both mixtures contain solutes of various polarities. In this experiment, various chromatographic techniques, that is, gas chromatography or the thin layer chromatography with spatial detection mode and bioautography, have been employed for determination of these two essential oil compounds. Juniper oil contains monoterpene alcohol. This led it require more polar mobile phase in comparison to thymic oil. This experiment cannot determine the purify of these separated zones, as the decreasing of the separation distances, simultaneously increasing the mobile phase polarity during the multiple plate developments, enhance the separation of the polar compound zones on one hand and keep non-polar compound zones separated on the other hand. This is the reference from our group. The article name is The New Thin Layer Chromatography Method for Separation and Determination of Multi-Component Mixture of Plant Extracts. This article is done by 2016. That's all from our group. Thank you.